This week on Bama Mysteries, a Birmingham student is killed while doing scientific research at her college campus. Why would someone want to kill an innocent student? Find out on tonight's episode. This episode contains adult situations and violence reenactments. Viewer discretion is advised. Monday morning, February 4th, 2019. 19-year-old Maria Curry awakes at her dorm at Birmingham Southern College on the city's western border. She knocks at her girlfriend's bedroom door and opens it. She realizes that 21-year-old Rosalind Franklin has not returned to her dorm. Maria runs to the science building on campus, worrying that she had stayed up through the night working on an experiment. In the research lab on the third floor, she finds her slumped over in a glove box. Believing that she is asleep, Maria attempts to awake her and finds blood. Shocked and afraid, she calls her friend Dalton, who urges her to call the police. After several minutes, both campus and local police arrive. Local authorities pronounce Maria's 21-year-old girlfriend as deceased. The shocked Maria is taken to the hospital. Affectionately known as Rosie by her family and friends, the student was known to frequent the Stevens Science Center. She was selected by her professor, Dr. Laura Stoltz, to assist in research for a new anti-cancer therapeutic. Rosie's hard work and professionalism earned her a spot as a presenter at the upcoming American Chemical Society convention. On the night of Sunday, February 3rd, Rosie left her dorm to resume working on the project. Before leaving, she informs her girlfriend of her night's duties and runs to the science building. Sometime that evening, Rosie hears a strange noise in the hallway. Believing that other students may be working in the other labs, she ignores the sounds and continues working. After a few minutes, a person appears behind her and fires a shot. Rosie is shot in the head and back, immediately killing her. Why would someone want to kill a high-achieving college student? Detectives are able to quickly discover that reason.
My name is Dr. Laura Stoltz, and I'm a chemistry professor here at Birmingham Southern. I was also the late Rosie, God rest her soul, research mentor, and we were working on a revolutionary new anti-cancer drug. We were collecting data to demonstrate the power of the drug at the upcoming ACS meeting. The night Rosie was shot, she was working on the drug in the glove box, and a sample of the drug is missing from my inventory.
Local detectives soon began looking into a 62-year-old University of Alabama at Birmingham professor. Dr. Gary Millarity was an associate of Dr. Stoltz. The pair had a major falling out over who truly discovered the new anti-cancer drug. Millarity believed that he and his UAB associates owned the compounds used to create the drug. Stoltz, however, argued that her group had proper evidence, including dated journals and lab experiments detailing the discovery of the drug.
So we've collected a lot of evidence, as you can see in our crime scene. We've already shipped that evidence off to our specialist. They will be examining it and testing it, giving our results back later. We have a lot of suspects. We have 4 right now at the time. We have a lot of different motives and a lot of different reasons for doing this. And so we look forward to seeing the results.
My name's Reid and I'm a drug specialist. So for this case, one of the experiments that we did was a thin layer chromatography test. So from the scene of the crime, we gathered an unknown white substance and we compared it to a bunch of 3 known substances. And so how we tested this was we dissolved them in water and placed them at the bottom of a thin layer chromatography plate. We put the substances at the bottom and then placed the plate in water, let the water rise to the top of the plate, then took the plate out and let it dry. Once it was dried, we were able to see how far the substances had traveled, and from there we were able to determine the RF value, which is the distance of spot traveled to the distance of the spot front. So from there we gathered that aspirin traveled this far, caffeine, acetaminophen, and our unknown. And based on the unknown, we had 2 spots, so we had to gather that it was 2 different RF values and get the average of both. And from this, we were able to gather that it was not the anti-cancer drug, but rather it was a mixture of caffeine and aspirin, showing that the substances that were found in the suspects did not match the anti-cancer drug.
I'm Mary Caroline, and I'm another one of our drug specialists. In order to further identify our suspects, we use the process of presumptive drug testing with the unknown substances found at the crime scene and on the suspects, combined with Mandolin, Marquis, and Scott reagents. Based on this chart given regarding colors that each known compound sample should turn, we compared the results of each unknown to the anti-cancer drug that Rosie was working on. Based on these results, we can tell that the unknown in the glove box that Rosie was working on is the closest match to the anti-cancer drug, which makes sense. However, the unknown from Maria and the unknown from Gary are not very close matches to the anti-cancer drug or the drug in the glove box, indicating that this test cannot convict Maria or Gary.
First thing each of our specialists did was collect soil from across campus, using a trowel and placing it into a bag marked and dated with location on campus. We then washed and separated our unknown and known soil samples into two categories, dried and not dried. The reason we dry the soil is to preserve its characteristics for as long as possible prior to testing. The first test we ran was an examination on the physical characteristics of the soil. This test we ran solely on the dried soil, as undried soil can show similar colors. This helps us distinguish the characteristics in the soil. Then we darkened the room and took our UV light to each of our samples, making specific notice of any unique colors shining or color intensity. From this, we concluded some matches appeared which show signs of promise, but further analysis is needed to make any conclusions.
The second test we ran was determining soil density. We did tests using cylinders to record the density and report the soil settling time for each sample for our known and unknown. Next, we made the calculation using correct sig figs and recorded our density. We concluded that the soil's density was closely related to Gary Malarity's shoe, but because of weather and different factors, this is not enough evidence to convict and we need further tests.
The third test was the phosphate test. Hi, I'm soil expert number 3, Mitchell Gibbar. Like I was saying, the third test was the phosphate test, and from this test, our expert was able to analyze the substance's color in relation to the other colors inhabiting the other wells. This enabled the scientists to effectively gauge the ppm phosphate concentration embedded in the soil. Furthermore, the scientists also had the means to compare the consequent colors of the unknown soil specimen with the colors of the known soil specimens. Lastly, we tested the flame color of the soil, using a unique lens that helped us see specific colors. From this, we concluded many matches, some promising, others leading to more questions concerning the killer, Miss Rosie. Right now, Gary Malarity is our prime suspect, but further testing will be needed to confirm our evidence.
After thorough analysis of the crime scene, we were able to recover a fiber specimen that was found in close proximity to the area in which the crime was committed. In order to determine the origin and the makeup of the specimen, we collected a field of fabrics and displayed various fiber attributes. First, we tried to identify the origin of the suspected sample we collected in order to analyze it from a macro perspective. We wanted to observe the physical characteristics of all our fiber specimens, such as color, thread count, size, and individual weave type. The initial analysis of the fibers allowed the team to be able to narrow down our comparisons based on our large-scale macro techniques.
After the analysis of the fibers and the evidence, this showed that the fiber samples taken from the crime scene did not directly match any of the specific samples that we took from the suspects. There was no direct match, although there were some similar characteristics. This fiber evidence is inconclusive, and we will do further testing to determine the suspect.
In the crime scene, we used some paper from Rosie's notebook that we believe would reveal some of the suspect's fingerprints. It is best to make use of the iodine chamber to reveal fingerprints on porous surfaces. We took pieces of the paper and placed them inside of a jar of iodine. After leaving it in for a few minutes, fingerprints began to appear on the paper. In order to create a more permanent view of the fingerprints, we sprayed a starch solution, which turned them from a brown color to a black-blue color. The darker color also helped us view the fingerprints better. The color created defined lines that we could use more easily to compare our fingerprint samples. We were able to identify Rosie's prints on her own notebook, but that is understandable. We also were able to match a fingerprint to Gary Malarity.
One way of finding prints is dusting for prints. The first step is to look at the crime scene with UV light and spot fingerprints. Once spotted, we grab a light brush, and dust around the surface with the substance called fingerprint dust. After the step is complete, the fingerprint should be visible. The next step is to grab a piece of tape and put the piece of tape down and lift the fingerprint up, and the print should be on the piece of tape. And then we can compare that to the suspect's print.
We collected bullet casings from the crime scene where Rosie was shot. We then prepared an acidified hydrogen peroxide solution. We used forceps to place the brass casings into this solution. As the casings were submerged, they started to turn a brownish green color and had sort of an oxidized look to them. Bubbles would form. We had to knock the bubbles off so that the space around the fingerprint could continue to form. We pulled the casings out of the solution and allowed them to dry. There were clearly fingerprints there, however, they were not visible enough to be compared to our suspect's charts. There were fingerprints there, however, they were not visible enough to be compared to our suspect's charts. The results of this experiment were unfortunately inconclusive.
After analyzing these gun casings for fingerprints, it was next important to analyze the gunshot residue left on Rosie's lab coat. There was an entry point in the back left that left a small amount of residue that could be tested to determine how far away the suspect was when firing the fatal shot. The results from this later can be linked to the multitude of footprints left in the lab that correspond with varying suspect shoes that we've collected throughout this research. To begin, we placed 2 thicknesses of paper towels down on a flat, heat-resistant surface. Next, we placed our sheet of test paper with the 2 chemicals on it on top of that. Afterwards, we placed our GSR specimen face down on the test paper. Following that, we saturated a paper completely with vinegar and placed that on top of the GSR specimen. Next, we set a iron to medium heat and placed that on top of it and let that rest for 2 to 3 minutes. Following that, we lifted it up and examined each one of the control specimens and compared it to the specimen on Rosie's coat and tried to determine which distance was actually fatally shot from. Following this procedure, we determined that the suspect was between 8 inches to 1.5 feet away from Rosie when she was fatally shot. This further can be used to determine which footprints will link these gunshot residues to the suspect.
So we're here at the crime scene of Rosie Franklin. Coming into the case, we had 4 suspects, those being Dr. Schultz, Maria, Dalton, Maria's friend, and Dr. Molarity. With the help of the evidence that our homicide team found, we were able to carefully narrow down our suspects to one in particular. Although some of the tests we performed were inconclusive, the soil test using the dirt found on the bottom of Dr. Molarity's shoe and that at the crime scene matched, and the fingerprint test that we did on the evidence found in the crime scene also matched Dr. Molarity. So those 2 pieces of evidence were enough to put him under arrest and convict him of the murder of Rosie Franklin.